Dissolution testing, dissolution method development, dissolution method transfer, and dissolution comparison. This is very vast topic, so I keep on making the videos on these topics, as dissolution technique, dissolution understanding, and its application is very vast. So we need to understand the topic in very much detail. I have made many videos, but still I am keeping on making the videos on this topic. I also request all the viewers to watch these videos repeatedly, so that you will get the in-depth understanding about this topic. Welcome dear viewers to this video. It is a part 1 for the series, and another part will be there. Soon I will be making another part. So both parts should be watched. Dissolution method development and profile comparison. So you can go with the QBD approaches for dissolution method development as a advanced technique. Earlier, the dissolution methods described in the monograph was used. It can also be used, but we need to show the discriminatory power whenever we are using the compendial method also. It is not like that if the method is in monograph or pharmacopoeia, we don't need to show the discriminatory power. It is not like that. We need to show the discriminatory power. Quality by design principles can be used for dissolution method development, and this video will provide you a practical consideration for developing and comparing the discriminatory dissolution methods. Also, we will see overview of discriminatory dissolution method, how to develop it, comparison of the profiles, and how you can select the right comparison method, and in last we will see the summary. So discriminatory dissolution method development is very important, as the regulatory bodies are requiring or asking for the report for dissolution method development. If you are using the compendial method, that time also you need to show the discriminatory power. Importance of formulation screening, quality control, and regulatory submissions. These are the key considerations for the discriminatory dissolution method. This method discriminates between significant formulation changes, process changes, and the stability. It is aligned with the QBD principles and life cycle management of the product. So stepwise approach is like check the USP method. If USP method is available or not, then check for the OGD method, FDA method, if it is available or not. If both these are not available, then you can develop its in-house method. You can also modify, or you can also change some parameters for the available method as per the product. And then step 4 is comparative dissolution testing and the profile comparison. Dissolution profile comparison. It is required to show the similarity between the dissolution profile of the two products, like test products, along with the reference products. Different strengths might be there. Sometimes during SUPAC changes, the pre-change and the post-change formulation is tested and compared for dissolution test, compared for the dissolution results. Also, multiple strengths are required to be checked. So for comparison, there are some methods provided, like F2 similarity, bootstrap F2 similarity, and MSD method. MSD method is multivariate statistical distance method. So differences in data needs a statistical rigor and when it is required to be used. We need to understand when these three methods are used, what are their advantages, limitations, and what justification is required. So F2 method is a average based or mean based method for dissolution profile comparison. It is simple comparatively to other methods. It is a regulatory default method, but it has a limitation for poor handling of the variability. If there is much variability, and as this method is mean based, so it becomes a limitation. F2 value above 50 means the profiles are similar. F1 F2 method, you might be aware about that. So this is the first method, that is called as F2 similarity method or F2 factor method. Second is bootstrap F2 method. So it is based on the confidence interval approach. It reflects inter and intra batch variability, and it is suitable for the borderline or variable cases. As we are aware about the F2 method requirement, the data variability at initial time point and later time point is required to be considered. The variability for F2 method is not more than 20% CV or RSD at initial time points and not more than 10% at later time points. So if the variability is more, then go for the bootstrap method, as bootstrap method provides the confidence interval approach. Then if F2 is also not suitable, then third method is MSD method. It is most rigorous and data intensive method. It evaluates profile shape across all time points. It is used when both the F2 method and bootstrap F2 method failing. That time MSD method can be used. So selecting the right comparison method is very important, and how to select the right method is very critical. Step 1 is to check the F2 method, whether F2 is above 50 or not. Second is check the variability is acceptable or not. If variability is higher, go for the bootstrapping method. Use bootstrap F2. If it is above 50, you can say that the profile is similar. If it is not above 50, if it is failing, then go to the MSD method, and MSD method will provide you whether the comparison is passing or failing. So scenario summary, like scenario 1, 2, 3 and 4 and 5 will be there. Scenario 1 is pass with F2. Scenario 2 and 4 means additional assessment is needed if the F2 criteria is failing, and scenario 5 is confirmed with the MSD method. QBD principles ensure robust and reproducible dissolution method development, and it helps to choose comparative dissolution profile methods based on the variability and the regulatory expectations. So we can start with simple F2 comparison, go to the bootstrapping method or MSD method based on the outcome and variability. This is a chart or decision tree for deciding whether to go for the bootstrapping method or MSD or not. So if in simply if your method is providing the F2 value, then it is passing, no need to go ahead. If the results are variable, then go to bootstrap, and if still bootstrap is failing, then MSD method is required. So if MSD method is also not working, then you need to go for the additional testing, additional dissolution method development, and IVIVC. So start is the F2. If F2 is matching, check the variability. If F2 is failing, if variability is within limits, then F2 is passing. That is the end of the dissolution profile comparison. If variability is not within limit, then go for using the F2 by bootstrapping. If F2 bootstrap F2 is passing, then stop there itself, and if it is failing, try for the MSD method. MSD confirms similarity, that is pass. If MSD is failing, that time it is a serious problem with the formulation or with the dissolution method. So if MSD fails, additional assessment is needed. So the product may be not similar in the dissolution, or some problems might be there in the dissolution method. So when both F2 method using bootstrap and MSD test is failing, then need to develop a in-house dissolution media or method which is discriminatory in nature, which is providing the biorelevant relevance, and it is specifically used for the test formulation along with the same testing of the reference formulation against which or in comparison of which you are going to show the similarity. So for this, use different combinations of dissolution factors like pH levels, media volume, rotation speed, or you can choose between the apparatus. So the objective will be of this activity like achieving the range of percent drug dissolved or release profile. Next step is to identify the media that shows the highest correlation with in vivo. So biorelevant method, discriminatory method is required to be developed. Then the goal is to establish a strong IVIVC, that is in vitro in vivo correlation, and align in vitro and in vivo drug profiles. So this is regarding dissolution method development and profile comparison. Second part of this video series will be coming soon. I request you to go through that video also so that you will have good understanding about this topic. Thank you for watching the video.